Hi everybody, it's me, Mr. Olson, and I teach you biology here at Mount Dora High School, and here I am virtually teaching you. Lucky me. Lucky you. Lucky us. Alright, so we are doing our Unit 5 review. The test is tomorrow, as you can see. That's December the 8th, so please be ready. These notes that I'm using are posted in your Google Classroom, and they should be found next to where you're finding this video. And there was a study guide that goes along with that, and of course, as you guys remember, I follow along with the study guide. So you have those posted. I posted them for you Wednesday evening, so they are live for you now. You can follow along, add notes if you wish, and of course you can just look at the review slides that we're using now. All right. So first off, we have the cell cycle, and the first question is, what is the cell cycle? Well, it is the regular cycle of growth and reproduction for a cell, or namely, it's just the life cycle of a cell. On the image on the right here, we can kind of see that's the donut of life, or that's kind of what I referred to it earlier. We said everything kind of starts right here. I hope you're seeing this. Everything starts right here, and the cell goes through these stages, basically like a clock, kind of working its way around from the very top of the clock all the way around. And the big orange bar that you see there is going to represent interphase. The blue bar and the red bar and the green bar are going to represent the different phases within interphase. We said G1 stands for gap 1, S stands for synthesis, and G2 stands for gap 2. So those would be major stages of the cell cycle. The final one here would be mitosis, and that is basically the cell actually dividing. And then it's actually followed up right there at the end, where my little cursor is going here. Right there at the end, it goes to cytokinesis. So it says, what are the most common stages of the cell cycle? Well, the most common one in general would be interphase, because it's like 95% of a cell's life, once again. But G1 is almost 50% of a cell's lifespan. And remember, this is a cycle of regular growth, copying more organelles so the cell can function as it gets larger and stuff like that. So most cells spend most of their lives in gap 1. It says what stages make up interphase? Well, we just kind of saw that. G1, synthesis, and G2 would be the stages that it takes place in. G1, of course, is gap 1, S is synthesis, and G2 is gap 2. Like I said, we've been reiterating that for a long time. You should know those are part of interphase. That's why that question's here, and you should expect maybe a question that makes you tell me exactly what things might be part of interphase. Next up, it says what occurs in each of those stages. Well, G1 is known for mainly growth, usually in the size. We did an assignment previously where it asked you a question, basically, how can growth be measured? And growth in cells can be measured in size and in numbers. Well, mitosis later on will increase the number of cells, but G1 and G2 really play a role in growing the size of the cell. So not only is it getting bigger, it's also making more organelles, and at the very end, right here, this plays a role with cancer later on, but right here at the very end of gap 1, there is a checkpoint that kind of signals to the cell, all right, you're healthy, you have the right size, you have the right components, it's time to go ahead and replicate your DNA. You can go through synthesis next. And that's what is next, synthesis. That's where we copy, or we double, or we replicate, however you want to phrase it. You make more DNA, and it's got to be an exact copy of that DNA that was there. This actually allows the cell to divide and still keep all of its key information in all the future cells it makes. Then finally, G2 takes place. Of course, more growth. It physically gets larger in size. Not a whole lot, but a little bit. It also prepares for division. Namely, it makes sure that some of the key components of cell division are there. And then it says there's another checkpoint. So right there at the end of G2, right before mitosis, there is another checkpoint that takes place. And this is kind of like probably the most important checkpoint. If this cell fails to pass that checkpoint, then that's a signal to the cell like, hey, I might have to self-destruct because I could be too dangerous to actually replicate. And then, of course, everything that follows would be, well, mitosis. All right, let's move on to the next. It does say what is gap 0. Well, gap 0 and what kinds of cells might be found in it. So gap 0 is kind of found off to the side over here. We basically said the cell just stays in gap 1. It doesn't grow, it doesn't develop, it doesn't go past go and collect $200. It just stays functioning normally. And that's good for some particular types of cells, namely brain cells. You don't want your brain cells dividing all the time, because that means like 5% of your cells at any given point in your brain wouldn't work normally. Whenever a cell's going through mitosis up here, they're too busy ripping themselves in half to worry about their mitochondria, their chloroplasts, their protein synthesis. It's not working the same way. So that's too risky. So the brain mostly stays in gap 0. Same thing's true of your liver cells. Pretty much any cell where we don't want any change, we don't move it past gap 1. So what we call it is gap 0. It says differentiate between sexual and asexual reproduction. Well, sexual reproduction means it needs a partner, and asexual reproduction means it doesn't need a partner. So everything we just been talking about for the cell cycle and for mitosis normally is considered to be asexual. Those cells can make more copies of cells without help from any other cell. They by themselves will make a copy. Sexual, of course, does need another cell to mix together to form a new combination of genes and make something more diverse. And that's what it says down here. Sexually is more genetically diverse. And that might sound familiar because that's a phrase we used for not mitosis, but meiosis later on. It does help in survival, namely keep species going. But asexual, no diversity in cells. We want exact copies. When I cut my finger, cutting some onions, cooking dinner, and I want to replace those cells with copies of those cells. I want those finger cells to be exactly the same. I don't want nothing weird in there. It's also pretty much the same as cloning, just naturally occurring, and it's very efficient. So all good things to remember and note. All right, so let's talk about mitosis. So some key information here is the abbreviation of letters that I gave you up here, IPMATC, stands for interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. You might remember my goofy little story that somebody was going to invite me over for dinner, and then I went over there, and then I made a mess on one of their bathroom mats. I peed on your mat. See? That was a story to help you remember those 6 letters in that order, because it's the stages of mitosis and, honestly, the whole cell cycle. Also off to the side, you're going to see it says somatic and body cells only. These somatic and body cells basically means all of your cells go through mitosis except for a couple, which might be either egg or sperm, and that's only meiosis. But we're talking about mitosis, and I want you to know that out of the gate. All of your body cells pretty much go through that process. So what happens during prophase? Prophase, the chromosomes, originally called chromatin, is kind of loose. It's kind of like loose spaghetti inside of a cell, and it's not distinguished anywhere. So those chromosomes, those start to condense, start to hug together, get really close, and that starts to make those X-shaped chromosomes that we saw earlier. Now we can see them very easily. The nuclear envelope also dissolves. The nuclear envelope just basically means the nucleus is going to dissolve. And then these spindle fibers form. The large spiders reach across, right? We talked about that before in a little story. And these spindles are basically long protein fibers that are going to actually help pull apart all of this stuff. Now, what happens during metaphase? Well, in metaphase, those chromosomes we just spoke about, they line up in the middle of the cell in a straight line. We actually called it the metaphase plate. So it's really easy to identify, especially in your labs that you guys had to do. And spindle fibers, they're going to officially attach to the chromosomes. And that's going to help lock them in and then ready to pull apart, because once again, the spindle fibers will do that action. What happens during anaphase? Well, it's the phase of mitosis where the chromatids separate. So we said 1/2 of a chromosome is called a chromatid. There are 2 identical chromatids that make up one chromosome. And these spindle fibers, when they pull in opposite directions on the cell, it splits that chromosome in half. So chromatids separate. And they do move to opposite ends of the cell, and some organelles will move over there too. Not really a big deal for us in our course, but hey, got to mention it. Worth saying. What happens during telophase? Well, after the chromatids separate, this means that the cell is going to form new nuclei. So we've kind of regrouped all that information together, and that's pretty much the final stage of mitosis. Now, we will go ahead and add in there, after telophase, that's when cytokinesis occurs. That's why that letter C is here, because there is the cutting of the cell cytoplasm, but more on that in a minute. It says, explain the job of the spindle fibers and the centrioles. Well, we've kind of already said it. The spindle fibers, they're going to do the work of pulling apart the chromatids, especially during anaphase and telophase. But the centrioles are kind of the anchors of where the spindle fibers kind of connect to the north and south pole of the cell, and basically are pulling the chromatids towards those areas. So the centrioles kind of are the center of attention in that regard. So here's an image that I found for us. I like it. I don't know how many different images we've used in our notes before, but it does kind of review a lot of the stuff we just mentioned. So remember, interphase does include kind of a looser interior to the inside of the cell, and then that one cell is going to go through the cell cycle, goes through G1, synthesis G2, and then it'll move on to prophase. Look how much more easily we're starting to see these chromosomes are going to kind of reshape and kind of take a thicker thread. That's the chromatin that we were talking about earlier. And then from there, we move on to prophase. In prophase, now it's like that classic X-shaped chromosome that we've been talking and playing with for a long time. So it says the chromatin condenses, making the chromosomes visible. And then those centrioles that we just talked about just a few seconds ago, those are going to be found on the edges of the cell structure, and then those long spindle fibers are going to start growing out from there. There is prometaphase. Hey, it's not going to show up on the test. We said we're going to ignore that stage because we mostly care about what happens in metaphase next. So those chromosomes line up in the center of the cell. We call that the metaphase plate. The centrioles are in position in the north and south poles. Those long spindle fibers have attached to the centromeres of the centrioles, and they're basically lining them up, getting them ready for kapow, anaphase. So anaphase is going to start separating the chromatids from each other. They're going to move in the opposite directions. The shortening spindle fibers is what's pulling them apart. And then telophase, we're basically going to have those chromatids kind of group back together. We'll start to reform the nucleus here on both of those. And you can even start to see in this picture, it does start to split that cytoplasm in the middle. Not totally there yet. That comes a touch later, but after that, we end up with two separate cells, and then that DNA can relax again. So this image up here is kind of later telophase, and this is more like early telophase, maybe late anaphase. But there you go. That's kind of the process that we've been talking about. And I know you've done this a few different times, but it's always worth reviewing in a review. I also want to point out to you, we're about to talk about chromosomes in better detail. I want to remind you that the cell is pretty big by comparison. Inside is the nucleus, and inside of the nucleus are those chromosomes. Remember, there's that thick thread, and that thick thread is basically the chromatin that's been wrapped up and arranged in this cool X shape. I will also go ahead and remind you that one half of a chromosome, one half of this X, is called a chromatid. So when we said the chromatids separate, we're saying this little junction right here, this little point, breaks, and then one half of this moves one direction, and the other half of this moves the other direction. We did mention that this is basically DNA. It's just wrapped around proteins, and that basically makes it thicker and a little bit more clear, a little bit easier organized for our cells to work with and separate. So continuing with the mitosis questions here, what's the difference between chromosomes and chromatin? Well, we just saw an image of it. Chromosomes are basically condensed and really highly organized DNA, whereas chromatin is basically just DNA wrapped around small proteins that we call histones, and there's also nucleosomes. But basically, chromatin is looser and it's stringier, so it doesn't have as much organization involved. Describe what occurs to the DNA during mitosis. Well, it not only gets condensed, which we've said half a dozen times already, it forms those chromosome shapes. It also gets separated at the centromere, so that junction between the two chromatids, that's what's going to break eventually. And finally, it gets loosened back up into chromatin when it's reforming its nuclei, namely during telophase. So a lot happens to the DNA. Before mitosis, that's when synthesis happened. That's when it's copied. And I have noticed, especially from the activity from yesterday, a lot of people made a mistake by saying, oh, we copy the DNA during prophase. No, that happens during synthesis. It says, what are the final products of mitosis? Well, the goal is to make two identical daughter cells. That means these two cells that we make, the original one's not there anymore, and its parts and pieces are used to make two brand new cells, but they're identical to the parent and identical to each other. There's also a note here that says both are diploid cells. That means that they have two sets of their genetic information so that they can kind of split over and over and over again. Final item here says, explain how cytokinesis is different from telophase. Well, cytokinesis, the kind of breakdown of the word here, cyto actually means cell. So cytokinesis is cell cutting, and that basically means we're splitting the cell's cytoplasm. Cytoplasm is the jelly-like substance that makes up a majority of the space inside of a cell. So if we're cutting that, well, we're working with two different cells. Telophase is only concerned on the nucleus and whatever's inside of it, not the cell's body. It's just the nucleus. So the DNA is the only thing we focus on. After telophase and cytokinesis do we actually officially have two brand new cells. And remember, they're copies to each other, no differences. If there's differences, there's problems. So meiosis, my big note up here says sex cells only. That means only sperm and egg are actually created through the process of meiosis. And this is a much more complicated sequence. So you remember previously it was I, PMAT, C. Now, well, PMAT gets a little bit more repeated. So it goes through interphase. It makes, G1, synthesis G2 happen. Then it goes to P1. So prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. So we do cut it into two cells, but that's the first stage of cell division, which we officially call meiosis 1. So everything that I'm kind of circling right here, that's meiosis 1. Then we go with those two cells, each of them go through another stage of division, which we call meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and then another form of cytokinesis. Notice, in between prophase and cytokinesis, we did not go back through interphase to do synthesis again. Technically, it is kind of an interphase. It kind of relaxes, but it doesn't copy its DNA. That's important because we want to reduce the number of chromosomes, not keep them. So anyway, after two stages of cytokinesis, that means we've made 4 total cells. This would be a good time to define what diploid and haploid actually mean. So diploid basically means you have 2 copies of each chromosome, and haploid, the type of cell made in meiosis, only has 1 copy of each chromosome. So we don't expect that to go on and make more copies of something. It's going to be used for another process. So what exactly occurs during meiosis 1, other than the stages which are already listed there? The first is basically it divides the cell's nucleus, and it will make 2 cells. So prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Hey, you gotta know, I'm gonna have questions on this test that directly ask you what's the stages in order of either process. Also, there is some very weird stuff that happens here. It's not like mitosis at all. We have 2 things that occur. First off, crossing over happens. If you guys remember in the activity on the table from yesterday and the day before, you guys were breaking off beads and reattaching them from one color to another. That represented crossing over. That's the mixing up of chromosomes just naturally before they even split, which helps create more genetic diversity. There's also a process called independent assortment that occurs. And that one's tricky, but generally what it means is that chromosomes don't care what other chromosomes do in terms of how they're arranged, which means when they segregate or divide, it doesn't matter which one's been where. Blue and orange were the colors we used yesterday. The blue ones can all be on the left, or the orange ones can be on the left, or different chromosome pairs can be in different orders. And that actually adds to more genetic diversity. Technically, this is talking about homologous chromosomes, so that means they're the same size. That's why your beads yesterday, you put the big chromosomes next to each other and the small ones next to each other. Now, meiosis II, that sounds more familiar. It is another stage of division, but it acts pretty much just the same way as mitosis did. So each of those 2 new cells from meiosis 1 are each going to divide, and those Chromosomes are going to line up like normal during metaphase 2 now, and that will split it eventually through anaphase and telophase into making 4 cells. So it goes prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. And also remember, that is where the chromatids actually separate. So the magnets do get pulled apart, like we saw yesterday. How is this process similar to mitosis? Well, meiosis II is basically splitting chromosomes, splitting sister chromatids from each other the same way mitosis did. Whereas meiosis I, that's actually separating homologous pairs. So kind of the twins and best friends kind of move away from each other. How is this process any different from mitosis? Well, big thing is, of course, it's making different numbers of cells, but also different types of cells are involved. We said gametes, which are sex cells. Up here it says sex cells. Down here it says gametes. They are exactly the same thing, which basically means sperm or egg. But mitosis makes copies of body cells. So meiosis only works with gametes and sex cells, but mitosis only works with body cells. And it does reduce the number of chromosomes. So instead of having 2 copies of each chromosome, we're only going to have 1 copy of each chromosome. And that's going to allow more diversity to build up later on. So this is a pretty clean image, and I do like it quite a bit. This shows the stages in order of meiosis, and they're labeled nice and clean for us. So prophase 1, we can see that we have those X-shaped chromosomes forming here. Now, these are homologous chromosome pairs. So I'm going to remind you again of your beads. And you can see they're kind of sticking to each other. Some funny little crossing over is going to happen. And so by the time we're in metaphase 1, we're going to see some really some exchange of material that happens here. So by the time they get pulled apart in anaphase 1, you can start to really clearly see different colors are mixed up in the different sized chromosomes. Once again, this leads to more genetic diversity, a very good thing for survival. And then we get the telophase 1 and cytokinesis. We can start to see the cutting is going to start kind of having the cell here. Those chromosomes are going to start kind of looking a little bit less than what they were before. We had 2 pairs side by side here, but now they look kind of more like our classic chromosome shape from earlier. You'll notice in this case we have 2 reds and 1 blue on this side, and then 2 blues and 1 red on this side with a little color mix up in there. Once again, creating more genetic diversity. For meiosis II, prophase 2 is basically going to loosen up that nucleus that's been reforming, and those spindle fibers are going to do what they do so well. This looks almost the same as what we saw in mitosis. They form that straight line, they split the chromatids so the 2 different sides are moving towards the poles of the cell. We basically close it off using a nucleolus, and we start to cut the cell, and hey, we end up with 2 more cells for each side. So that means there will be a cell there, a cell there, a cell there, and a cell there. So we end up with 4 unique haploid cells. None of the interior of any of these cells look the same to each other. If you look at the size and color differences, they are significant. So we make unique cells. And that's a big deal, really important. Continuing the idea of meiosis, what are the final products of meiosis? Well, the phrase we would say, and really the goal of meiosis is it makes 4 genetically unique haploid cells. Not diploid, haploid. They only have half the number of chromosomes you would expect from a diploid cell. So there's that tricky one. What is independent assortment? Well, it's basically the random distribution of pairs of genes on different chromosomes to the gametes. Basically, we get to move the chromosomes however they want, left side, right side, and it doesn't matter what other chromosomes do. They each get their own choice. Now, for all of our examples we've been talking about, we've only been looking at 2, 3, maybe 4 chromosomes. But for human beings, we have 23 pairs of those chromosomes. So there's a whole lot more diversity in there. And remember, independent assortment basically is determined through metaphase 1 and anaphase 1. That's when that separation will take place in anaphase, and they'll kind of arrange themselves in metaphase 1. Not 2, 1. What is crossing over? Well, this is when the homologous chromosomes hug each other, get so close to each other, they basically mix up some portions of their chromatids during the process of meiosis. So this mainly happens during prophase 1 and metaphase 1. You can tell basically all of Meiosis I, a lot of key things really happen. So that's where a lot of money is made. Not actually, but a lot of diversity is made. How do chromosomes act differently during meiosis as compared to mitosis? Well, remember, in metaphase, and that's part of mitosis, all the chromosomes have lined up in the middle of the cell. No side-by-side business, no best friends hanging out with best friends. Each separate X-shaped chromosome form a straight line, and then those sister chromatids pull apart. In metaphase 1 of meiosis, homologous chromosomes line up. That means 4 separate chromatids are side by side. And basically, they exchange some information, so crossing over definitely happens. In metaphase 2, the sister chromatids finally pull apart. So metaphase and metaphase 2 are very similar to each other, but metaphase 1, pretty different, pretty unique. All right, so after we talked about mitosis, we actually jumped ahead and talked about cancer. And so we want to talk about some of those things just generally. We can't talk about exact causes for everything because there's just as many causes as there are types of cancer, but we had a better understanding of what cancer was based on what we know about the cell cycle. So what is cancer? In short, it's unregulated cell growth. That means the cell is continuing to grow even though it shouldn't. It should be checked and basically identified by the body and say, hey, you're not doing what you're supposed to. You need to go away. Those cells are allowed to persist, and that can form basically a tumor in different types of cancers. So how is it related to the cell cycle? Well, basically, in most cases, the cell cycle is severely shortened, which means the cell is going too fast and not being checked. In a lot of cases, it's making some significant changes, but typically they're very small cells and they're rapidly dividing. And so mistakes not only are getting made, they're getting multiplied because the cell's not getting basically regulated to the point of saying, don't make any more. If it's self-destructed, that would solve the problem. By the way, when I say self-destruct, the real term I should be using is apoptosis. This is basically a pre-programmed cell death designed to destroy really messed up cells. So anytime a mistake happens, this is supposed to happen. But in cancer's case, it's not. So those cells just keep multiplying and replicating the mistake, and then of course, we end up with complications. Now, what do those checkpoints do for a cell's development? Now, we said previously there's a checkpoint at the end of gap 1, gap 2, and there's even a kind of a hidden one in the middle of mitosis. But basically, their job is to look for mistakes, look for certain chemicals that are in the right numbers, and cause apoptosis to happen if those mistakes are noticed. But of course, in cancer cells, they're blowing right by those checkpoints, so those mistakes are allowed to continue. So what about a mutation? We're going to talk more about these, especially in Unit 7, but a mutation is essentially a change in a cell's DNA. Now, in the case of cancer, it's bad. It's not a good thing. But there are some mutations that aren't dangerous, but in the context of everything we're mentioning here, I want you to remember it's a change in the DNA that allows it to sneak past the safety regulations and in turn causes all the damage. So what are some common modes of treating cancer? Well, the first thing that's probably going to happen if you go to a doctor and they say, hey, you have a malignant tumor, or you have cancer that's spreading, it's metastasizing. The first thing they want to do is basically identify where it is, and then they want to remove it. So surgery is the first option. Find it, cut it out, get rid of the big problem. There's still probably going to be a little bit of those cells left behind, and they could be spreading. That's what we said malignant means. Malignant cancers spread. So they'd probably follow it up with a combination or one of these, which is called radiation therapy. Basically, they hit you with some version of light that's considered radiation, and it's going to destroy cells' nuclei, which breaks up their DNA. Well, if we target bad cells, they won't multiply. Downside, we're probably going to hurt some healthy cells too, so there's some risk with that. And the same thing's true of chemotherapy. This is using medications, drugs of different kinds, and their job is basically to target cell replication. So on a chemical level, can we stop cells from copying themselves? And if that's true, we can probably stop cancer cells from making so many copies, give our body a chance to fight it off. Problem, we're also affecting healthy cells. So that's why people might lose hair, get nauseous very easily, might have immune system problems. So it's kind of a risk. It's definitely a risk. But we're getting better and better at being able to do this well. And then what are some things that might cause cancer? Well, carcinogens, things like smoking, asbestos exposure, radiation exposure, they can all play a role in causing cancer. They can cause those mutations and changes in DNA which cause that cell to skip those regulatory activities inside of the cell. Genetics can play a role too, not a guarantee, and family history can play a role, which is kind of related to genetics. But if you have a family history or genetics that say, hey, you have a higher chance of cancer, maybe start avoiding some of those carcinogens. And in general, don't smoke anyway. It's going to probably lead to cancer. But anyway, the key thing is mutations happening in those regulatory genes, which you don't have to know about what the genes are, but you got to know it affects DNA in a bad way. And finally, just some weird little things that we mentioned. There's a process called binary fission. You and I do not go through binary fission. Our cells go through mitosis in the cell cycle. But if simple cells like bacteria, namely prokaryotes, they're going to divide, they use that simple version that basically they just kind of rip themselves in half. They copy their DNA and they rip themselves in half. There's no spindle fibers. There's no cool organelles. They just kind of split. And it doesn't take as much time as mitosis. It's actually really efficient, and they do a good job. But, well, they don't have as much information inside of us as we do. So they're able to go through that process much faster. If our cells take 24 hours to go through mitosis, those cells only take 20 or 30 minutes. So it's pretty impressive. Budding is a funny way for cells to make a new copy from an organism that they basically make another copy, like a little conjoined twin. They clone themselves, it grows off of them, and then it breaks off and it lives its own life. But it's basically an identical twin to whatever the parent was. And then there's also fragmentation. So if you've ever seen an organism, we talked about starfish, if they ever lose a leg and it's not eaten or something, and they just have the leg floating on the ground, that leg can actually turn into a copy of the starfish. It's pretty unique. But yeah, this is typically only going to be found from larger organisms, whereas budding is usually a lot smaller organisms. But all weird, but all pretty cool ways that cells can make more copies of themselves and affect the life of an organism. And that's it, guys. That's the whole review. If you have questions, typically I'd answer them here, but sorry, I may not be there. I usually do ask you if you have any last-second questions before we take our test, so this would be the time to do that tomorrow when you're taking your test. But I will just say, have some confidence. You guys have done this practice quite a bit, and I think you'll do just fine. Remember, test corrections are also always an option. I will go ahead and tell you the topic and theme of your written question for your test. Remember, of the 100 points of your test, 10 of those points come from one question. That's a written question at the end. And the topic and theme will be basically describing what occurs during the cell cycle and mitosis. Surprise, surprise. So you should be as prepared as you can before that, know what goes on, and be able to talk about exactly those stages in order. That should be easy for a lot of you, and I hope to see a lot of 10 out of 10s. So remember, study these notes, look through your study guide, do whatever it is you need to do. But I hope this worked. I hope you heard everything okay. And hey, like I said, if you got questions, I'll see you tomorrow. Ask me then. But in general, I think you got this. All right. Bye, everybody.